Okay, so the topic of this presentation today is cellular respiration. You might know that cellular respiration is a process performed by the mitochondria in order to create a molecule called ATP, adenosine triphosphate, and that's what you see pictured here. Well, let's go ahead and get started. So when we look at adenosine triphosphate, you'll often hear it called the energy molecule of the cell. An analogy that you might also hear is it's kind of like the cell's money or currency. You know, the more money you have, the more goods and services you can purchase. Well, if you look at it in this sense, the more ATP that the mitochondria produces, the more work can be done. And here's a list of reasons why ATP is needed. ATP is required in movement. You know, you might be taking these notes right now. Well, energy is causing your muscles to move across your paper in a very coordinated manner, I hope, so you can read legibly your handwriting. The ability to reproduce requires ATP. You might know that sperm cells, male sperm cells, swim through the female reproductive system, and that swimming process requires the use of energy. In order for proteins to be created, protein synthesis requires the use of ATP. So this is just a small list of reasons why the cell needs to make ATP. So first of all, it's called ATP, adenosine triphosphate. You can see I've just highlighted the three phosphates in this molecule here. And around each phosphorus are the oxygens there. And so you can see there are three of them in this picture. And during the hydrolysis of ATP, if you recall, hydrolysis is where you take the addition of water, a large molecule with the addition of water and various enzymes can be broken down into smaller molecules. So ATP with the addition of water can be broken down into ADP and a single phosphate. That releases energy, usually in the form of heat. Now, if you're wondering in my notes, why does it say P with a lowercase I? Why is there a subscript I? You might often see that. That's just a reminder that phosphates are inorganic. Inorganic means they do not contain carbon. If you look in the picture, look at the phosphates in the picture, there's no carbon. Phosphates are inorganic. And so when we look at this simplistic diagram here of an ATP molecule, water plus various enzymes during the process of hydrolysis, the hydrolysis of ATP, water plus various enzymes will cause that third phosphate to be broken off, and in that process, heat, energy is typically released. And so it's this heat, this energy that's released, which drives cellular processes. Heat often activates enzymes, which start chemical reactions. So when we look at cellular respiration, here's the chemical formula for cellular respiration. In the picture here, you have an electron microscope picture of a mitochondria. I've heard students often compare this to kind of a footprint on the moon. That's a pretty good analogy. And so here's the mitochondria, and the mitochondria performs the process called cellular respiration. And so cellular respiration primarily occurs in the mitochondria. I say primarily because there's this early step known as glycolysis, which takes place in the cytoplasm of the cell. So in this artist drawing, we can see that inside of the mitochondria, there's an inner layer that twists back and forth. It's kind of colored an orangish color, and filling that inner orange layer is kind of a blue fluid like you see in this picture. This is called the matrix. That's going to be important as we go through the process. So cellular respiration begins with a molecule of glucose, the glucose which you obtained from the foods that you eat. So energy from the glucose that we eat is converted into you might know into 36 to 38 ATP molecules, depending on the conditions. Certain environmental conditions will cause 36 ATPs to be made. Certain environmental conditions can cause 38 ATPs to be made. But one thing I want to mention is that cellular respiration is very much necessary for both autotrophs and heterotrophs. You know, there's a myth out there that because plants are the only ones that have chloroplasts, then that means animals must be the only ones that have mitochondria. And that's a myth. That's not true. All eukaryotes have mitochondria. Eukaryotes include fungus, protista, plants, and animals. So mitochondria are essential in both heterotrophs and autotrophs, plants and animals, and fungus and protista to be exact. Okay, as we now get into the process, let's first examine what's called glycolysis. Glycolysis begins in the cytoplasm of a cell, and it begins with a molecule of glucose. You know, in my picture, I've shown glucose as the blue ring of circles. Notice how there are 6 carbons in that ring because there are 6 carbons in glucose. There's also 12 hydrogens and 6 oxygens. You know, for simplicity, I've only drawn the glucose as the 6 carbons. Just keep in mind there's still 12 hydrogens and 6 oxygens. So glycolysis begins with the addition of 2 ATP molecules. Now this might seem counterproductive at first. After all, the purpose is to make ATP. So why is ATP being spent? Well, the analogy I like to make is in business. You have to spend a little bit of money in business to make a lot more of money. For instance, you might have a wonderful invention that you think is going to make you very rich. But if nobody knows about the invention, no one's going to buy it. So you spend maybe a few $1,000s on advertising, radio ads and television ads, internet ads. You basically market the product. Spend a few $1,000s, and then to attract customers who will hopefully buy your product to make you millions of dollars. In that case, it's smart to spend a few $1,000s in order to make a few million. Well, in the production of ATP, in order to make 36 to 38 ATPs, which is what we're going to have at the end of this process, 2 ATPs are needed to start this process. So here's 2 ATP molecules. And with the help of various enzymes, ATP is brought over to the glucose, and with the help of various enzymes, the ATP is broken, heat is released. Notice how the phosphate was left behind. Well, there's a second ATP molecule there, and again, enzymes bring the second ATP molecule over to the glucose, break apart the ATP, heat is released. The releasing of the heat causes that glucose molecule to break into 2 fragments of molecules that we now call phosphoglyceraldehydes. Notice how phosphoglyceraldehydes are 3 carbon molecules. You might often hear me call them PGAL. PGAL is just an abbreviation for phosphoglyceraldehyde. Well, what happens next is, again, various enzymes. There's all kinds of enzymes in the cytoplasm of the cell. Various enzymes add a couple more phosphates to the PGALs. That changes them a little bit. They're not called PGALs anymore. And as the process continues, a molecule by the name or by the abbreviation of NAD. NAD stands for a molecule called nicotinamide adenine dinucleotide. I'm just going to call it NAD. Watch what NAD does. NAD acts like a taxi for hydrogen. NAD will pick up a hydrogen and deliver the hydrogen to the mitochondria. If you missed it, here comes another one. Another NAD will pick up a hydrogen and carry it away to the mitochondria. That NADH that was carried away, that NADH is going to be very important when we get to later parts of cellular respiration. Near the end of glycolysis, ATP is now also created. And so an ADP, with the help of various enzymes, will strip off one of the phosphates. Another ADP, with help of various enzymes, will strip off a second phosphate. Another ADP, with the help of various enzymes, will strip off the third phosphate. And yet another ADP, with the help of enzymes, will strip off the fourth phosphate. What's left behind are 2 3-carbon molecules that we call pyruvates. There's the formula right there for pyruvates. Notice how there are only 3 carbons to them. Those pyruvates are not waste. They have a role to play in the next step of they have a role to play in the next step of cellular respiration. But before we go into the next step of cellular respiration, let's just take a look at what's been created. 2 pyruvate molecules which they're not waste. Those pyruvates have a role to play still. A total of 4 ATP molecules have been created. You might often hear it called a net gain of 2 ATPs. Well, that's because 2 ATPs were needed at the start to produce 4. So when you take into consideration the 2 that were used at the beginning to make a total of 4, that's often what we mean by a net gain of 2 ATP molecules. And then notice the NADH molecules were created as well. Those NADHs that moved into the mitochondria. I like to call them a hydrogen taxi because they're delivering hydrogen from point A, the cytoplasm, to point B, the mitochondria. Kind of like a taxi delivers people from point A to point B. Let's move on. So now when we take a look at our cell, we're going to move to the powerhouse. Those 2 pyruvates are going to enter the mitochondria. So now what we need to do is we need to zoom on into the mitochondria and take a closer look. When we zoom on into the mitochondria, we can see the 2 pyruvates inside of the mitochondria. There's a layer in the mitochondria that twists and folds back and forth. That layer you might often hear is called the cristae. And inside of that layer, inside of the cristae, is a fluid called the matrix. And so the pyruvates are in that matrix area in the inner layer of the mitochondria. So when we now move on into cellular respiration, we're going to begin with the process called the Krebs cycle. You know, if you notice from the animation, again, the Krebs cycle takes place in the matrix. If you're in my biology class, there's a textbook diagram that's broken into various stages. And so when I say step 1, I'm also referring to the diagram in your textbook. So step 1, the pyruvates are broken into acetic acid. Well, you can see the 2 pyruvates right there. We're going to follow the blue pyruvate on top. The same thing happens to the one on the bottom, but for simplicity, we're just going to follow the one on the top. So what happens is, in order for the pyruvate to be broken into acetic acid, a few things need to happen. First, NAD comes and strips away a hydrogen and carries the hydrogen over to another area of the matrix. Those hydrogens are going to be really important when we get to the final step of cellular respiration. So in the process of the pyruvate being broken into acetic acid, carbon dioxide waste is also generated. There are enzymes that cause these bonds to break, and one of the carbon drifts away with 2 oxygens in the form of carbon dioxide. That leaves behind acetic acid. The 3-carbon pyruvate was broken into a 2-carbon acetic acid. One of the carbons exited as carbon dioxide. Well, now what happens with the acetic acid? So what happens next, we're looking in area 2 of the diagram from your book. A big molecule by the name of Coenzyme A is going to bond to that acetic acid. And when the acetic acid and the coenzyme A bond together, you just kind of mush the 2 words together. It goes by a new name of Acetyl-CoA. You can see acetyl-CoA is a really big molecule because the CoA, the coenzyme A, is a really big molecule. Well, what happens is this acetyl-CoA is a fairly short-lived molecule. It doesn't last very long. You'll see what happens in just a moment. When we move on into area number 3 of our diagram, citric acid is formed. Citric acid is formed when a 4-carbon molecule from the previous round of the Krebs cycle. Keep in mind the Krebs cycle is a continuous process. So from the previous Krebs cycle, a 4-carbon molecule is created. That 4-carbon molecule bonds with the acetyl-CoA. Enzymes break off that coenzyme A part, and that's the big part. When enzymes break off that coenzyme A, what's left behind is a 6-carbon molecule by the name of citric acid. So what we're seeing is the molecules change as a result of various enzymes. When we move on into area 4 of the diagram from our textbook, what we're going to see is that 6-carbon molecule by the name of citric acid is broken down into a 5-carbon molecule. Well, in order for that to happen, there are, of course, various enzymes, but also a molecule of NAD comes and strips away a hydrogen. The hydrogen accumulates again in another area of the matrix. We're going to see why that's important in a few moments. But the citric acid, which is a 6-carbon molecule, is broken into a 5-carbon molecule as a result of NAD stripping a hydrogen, as a result of enzymes, and also one of the carbons is turned into carbon dioxide. Citric acid is a 6-carbon molecule, but one of the carbons turns into carbon dioxide, and enzymes break it away and it floats away and it exits as waste. So now there's only a 5-carbon molecule left over. Well, what happens next? Now we move on to area 5 of the diagram. That 5-carbon molecule, with the help of various enzymes, is going to be broken down into a 4-carbon molecule. And just like last time, one of the products that's created is NADH. A molecule of NAD will come and strip a hydrogen away. The hydrogen accumulates with all of the other hydrogens in another area of the matrix. We're going to see why that's important in just a moment. Another thing that happens is a molecule of ATP is created. So an ADP and a phosphorus are brought together with the help of various enzymes, and that helps to bond ADP and P together to make a molecule of ATP. So another molecule of ATP is created. Now keep in mind, my notes say that 2 ATPs were made, even though I just showed 1, because we're following the blue carbons on top. The same thing happens to the blue carbons on the bottom. Just for simplicity, we're only focusing on the blue carbons on top. But another ATP molecule would have been created in the blue carbons in the bottom of the picture as well. So that's why my notes say 2 ATPs are created. 1 ATP from the top of the blue carbons on top, 1 ATP from the blue carbons on the bottom. And notice how my notes say a 5-carbon molecule is broken into a 4-carbon. Well, where'd the extra carbon go? Just like the last couple times, it's released as carbon dioxide waste. Enzymes break off one of the carbons in the form of carbon dioxide, and it's released as a waste molecule. Well, now we move on to area 6 of the diagram from our textbook, and it says enzymes rearrange that 4-carbon molecule from one 4-carbon molecule into another 4-carbon molecule. Watch the animation. 3, 2, 1. Enzymes rearrange the 4-carbon molecule from one 4-carbon molecule to another. In the process of doing this, a couple more hydrogen taxis are created. A molecule of NAD comes in and strips away a hydrogen, delivers the hydrogen to another area in the matrix. We'll see why that's important. And another molecule by the name of FAD, very similar to NAD. FAD comes and strips away not one, but 2 hydrogens. That's why it's called FADH2. All these hydrogens are accumulating in another area of the matrix because they're going to be used in the final part of cellular respiration. And so as we finish up, keep in mind that this is a cycle. In order for this to be a cycle, it needs to restart. So that 4-carbon molecule is going to bond with an Acetyl-CoA. We saw Acetyl-CoA a moment ago. The CoA will be released, and what's left behind is citric acid. Now we've restarted the Krebs cycle. And that's why it's called a cycle, because it's a continual process. And so the process simply repeats itself. Now, if we do a quick summary of the Krebs cycle, you can see that only 2 molecules of ATP are created. But the main prize from the Krebs cycle is all the creation of NADH and FADH2. Those are going to be hugely important during the final step of cellular respiration called the electron transport chain. Let's finish that up right now. So as we look at our mitochondria again, what we're about to do is we're about to zoom in deeper into the mitochondria. And what we're going to look at is the matrix, the boundary, the inner membrane separating the matrix, which is the light orange, from the outer layer, which is the dark orange. Let's zoom on in. All right, and when we zoom on in, we can see that embedded in the inner membrane is a protein channel, an enzyme by the name of ATP synthase. Now, if you've already learned about photosynthesis, you've seen this enzyme ATP synthase before. So it's going to be really important as we go through the next step. So if you are in my biology class, again, look at the picture. The picture from your textbook has numbered areas. So if we look at area 1, NADH and FADH2 are delivered, and again, their hydrogen is delivered. The NADH and the FADH2 from the Krebs cycle, and some NADH from glycolysis. So NADH from glycolysis or from the Krebs cycle is delivered. Well, watch what happens. 3, 2, 1. When the NADH delivers the hydrogen, the electron from the hydrogen is pulled to the inner membrane. The proton of the hydrogen, the H+ ion, is in the matrix. And this process happens repeatedly. NADH drops off a hydrogen, and the hydrogen's electron goes to the inner membrane, and the H+ ion is in the matrix. And again, we have another NADH dropping off the electron to the inner membrane, and the hydrogen ion to the matrix. And let's not forget FADH2. So here comes FADH2 delivering 2 hydrogens and 2 electrons. So what happens next in area 2 of the diagram? In area 2 of the diagram, those hydrogen ions in the matrix, the hydrogen ions at the bottom, are going to be pumped out of the matrix. And what we're setting up, or what's being set up, is a steep concentration gradient in the top of the diagram, outside of the matrix. A large amount of hydrogen is at the top of the diagram. A very steep concentration gradient is being established. That's going to be really important. When we move on into area 3, when the concentration gradient reaches a certain level, ATP synthase is activated. What happens is a hydrogen will pass through the ATP synthase. ATP synthase is an enzyme. And what ATP synthase does is it draws an ADP and a P together, allowing them to bond to make a molecule of ATP. And this process happens 32 to 34 times. Now I only show it, I think, 3 times in this animation. So a hydrogen diffuses through the ATP synthase that draws an ADP and a P together to create ATP. And the process happens again. Another hydrogen is drawn through, another ADP and P are brought together to make ATP. Now again, this can happen 32 to 34 times depending on the conditions. Now one of the problems that's happening is as more and more and more hydrogen passes through ATP synthase, the concentration gradient lessens. The 2 sides of the membrane are getting closer to equilibrium, and that's no good. There has to be the steep concentration gradient needs to be maintained, or ATP synthase stops working. So something has to come and pick up and remove those hydrogens that are in the matrix. And that will summarize area 4 of our diagram. Oxygen is going to pick up 2 of the hydrogens that have already passed through ATP synthase and a couple of electrons from the inner membrane to create a molecule of water. The water simply exits as waste. And as more water exits, taking more hydrogen, the steepness of the concentration gradient is reestablished. Now, again, in my biology class, you are hopefully preparing yourself for this essay question. So I'll talk to you about this essay question during class. And so there you go. If you're in my biology class, pause the video and try to put these steps in order from start to finish. If you're in my class, I'd love to see you before or after school if you want me to help you check your answers. So pause the video and good luck.